Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine diseases like Parkinson's and Alzheimer's. They slowly erode a person's ability to think, to move, just to be themselves.
Yeah, it's truly devastating.
And what's been so incredibly tough is the lack of real treatments, ways to actually stop that destructive cell death happening in the brain.
Exactly. There's been treatments for symptoms, but not to halt the progression.
But what if we're maybe on the verge of a breakthrough, like a way to actually pull the emergency brake on that runaway cell loss?
Well, that's the exciting territory we're exploring today. We're diving deep into a really promising discovery, a potential method to block cell death.
Which could lead to brand new therapies for these, heartbreaking conditions.
That's the hope, a big hope.
Exactly. So we've got to look at a health news story that flagged this up, and we're pairing it with the detailed scientific paper itself, published in Science Advances.
Right. So we get both the overview and the nitty-gritty details.
Our goal today is really to pull out the essential insights from both these sources, to understand this potential game changer in stopping cell death and what it could really mean for future treatments.
Okay, so let's get to the heart of this research. Scientists at the Walter and Eliza Hall Institute, WEHI, including experts from their Parkinson's Disease Research Center, they had a very specific aim. Yeah. They wanted to find small molecules, tiny chemical compounds, that could prevent cells from dying. The ultimate goal was paving the way for new treatments for those neurodegenerative diseases we mentioned.
Right. Okay, so we know that programmed cell death, it's also called apoptosis. That's actually a normal, healthy process in our bodies, usually.
Yes, it's essential for development and removing damaged cells.
But in conditions like Parkinson's and Alzheimer's, this process, it kind of goes into overdrive, right? Causing brain cells to die off too early.
Precisely. It's excessive unwanted cell death in the wrong place.
So the researchers were essentially looking for a switch, something to dial back this excessive cell death, specifically in those situations.
That's a good way to put it. And to do this, they use a really sophisticated method. At the National Drug Discovery Center, which is part of WEHI, they ran what's called high-throughput screening.
High-throughput screening. Sounds intensive.
It is. Think of it like testing a huge library. They tested over 100,000 different chemical compounds.
Wow, 100,000.
Yeah, just screening them all to see if any could stop cells from undergoing apoptosis from dying.
That's an enormous task. How did they even narrow it down? What were they looking for?
They were specifically looking for molecules that could interfere with proteins already known to be key players in the cell death process.
Okay, targeting the known culprits.
Exactly. And to make the search even more precise, they used specially engineered human myeloma cell lines. These weren't just any cells.
How are they special?
They were created using CRISPR-Cas9 gene editing, a very precise tool. These cells were engineered so that their survival depended on blocking just one of two key cell death proteins. One protein is called B-cell lymphoma. Two associated X protein. Let's call it BAX for short.
BAX, got it.
And the other is B-cell lymphoma 2 antagonist killer, known as BAK.
BAX and BAK. Okay.
So they had one set of cells where only BAKs could trigger cell death, and another set where only BAK could do it. This allowed them to see if a compound affected one pathway specifically.
That's really clever. It isolates the effect, doesn't it? So what did they discover from this massive screening?
Well, through all that testing, they hit on a particular small molecule. They named it WEHI 3773.
WEHI 3773.
Oh, okay. And this compound showed a specific ability to block the cell death that was being driven by BAK.
Only BAK.
Primarily BX in this initial screen, yes. They then did a lot more tests, obviously, to isolate this compound properly and confirm just how effective it was at keeping those BX-dependent cells alive. They measured things like cell viability and checked for damage to the mitochondria, the cell's powerhouses.
Okay, so WEHI 3773 is the start here. How does it actually work? What's it doing to stop BAK?
Right, the mechanism. The research showed that WEHI 3773 works by preventing BAX from moving to the mitochondria in the first place.
Oh, interesting. So it stops it getting to where it needs to do the damage.
Exactly. See, for BAX to actually trigger cell death, it needs to travel to the outer membrane of the mitochondria. Once there, it changes shape and essentially pokes holes in the mitochondria, causing them to leak and the cell to die. WEHI 3773 stops that initial move.
And how does it block that movement? What's the specific interaction it's disrupting?
Okay, this is where it gets really interesting and quite specific. WEHI 3773 achieves this blockage by interfering with the interaction between BAX and another protein. This other protein is already sitting there on the mitochondria.
Another protein involved.
Yes, it's called voltage-dependent anion channel 2. Let's call it VDAC2.
VDAC2. Okay.
It seems that BAX normally needs to connect with VDAC2 on the mitochondrial surface to properly embed itself and start the damage process.
Like a docking station.
Sort of, yeah. Think of it like BHs needs to plug into VDAC2 to get activated or positioned correctly. And WEHI 3773 essentially gets in the way, throws a wrench in the gears, preventing that BHE-DDA2 connection.
So WEHI 3773 is like a molecular roadblock specifically for the BHA2 interaction. Now, you mentioned BAX and the other protein, BAK, earlier. Does this compound affect BAK at all?
Yes, it does. And this is a really fascinating twist in the story.
Oh.
While WEHI 3773 inhibits BAX by blocking its interaction with VDAC2, it actually seems to have the opposite effect on BAK, at least under certain conditions.
The opposite effect? How so?
By interfering with BAK's interaction with VDA2, because BAK also interacts with VDAT2 WEHI 3773, can sometimes actually promote BAK-driven cell death.
Wow. Okay, so it stops BAX but can encourage BAK? That seems complicated.
It is. It highlights how finely tuned these processes are. This single molecule has a very different, almost opposite effect on these two closely related proteins, depending on how they're interacting with VDAG2.
That sounds like a really delicate balancing act inside the cell. So what happens in normal cells, which usually have both BX and BAK present?
Good question. Generally, in cells that express both proteins, adding WEHI 3773 tends to lead to increased cell death overall.
Increased, even though it inhibits BX?
Yes, because of a sort of feed-forward loop. Once BAK gets activated, perhaps helped along by WEHI 3773 interfering with its VDA2 interaction, the activated BAK can then go on to trigger BAK's activation directly in a way that doesn't even need VDA2 anymore.
Ah, I see. So activating BAK bypasses the need for the BAK's VDA2 interaction that WEHI 3773 was blocking.
Exactly. It shows how interconnected these pathways are. You tweak one part, and it can have knock-on effects elsewhere.
That's a really important point. It underlines the complexity.
It does. And the researchers also found another potential application linked to this BAK effect. They tested WEHI 3773 on leukemia cells that had become resistant to a cancer drug called venetoclax.
Venetoclax. Okay. How does resistance happen?
Well, sometimes cancer cells become resistant by losing their BAX function. If BAX isn't working, venetoclax, which relies on BAK's dig pathways, might stop working too.
Right.
But they found that WEHI 3773 could overcome this resistance in BAK's deficient leukemia cells. It did this by promoting that BAK-mediated killing we just talked about.
So even if the cancer cell shuts down the BAX route, this compound might be able to force it down the BAK self-destruct route instead.
That's the idea. It's like opening up an alternative pathway to kill the cancer cell when the primary one is blocked. A potential strategy against drug resistance.
That's really quite clever, a potential workaround.
Precisely. And just to be really sure about the mechanism, they use techniques like drug affinity responsive target stability assays, DARTs for short.
DARTs.
Yeah. These tests basically confirmed that WEHI 3773 physically binds directly to the VDA2 protein.
Okay, so it definitely targets VDG2.
Yes. Both when VDAC2 is on the mitochondria in cells and even when they use purified VDC2 protein in a test tube. They could even pinpoint the specific region on the VDAC2 protein sort of beta sheets, 7 to 10, if you want the detail that's crucial for WEHI 3773's activity.
That's incredible detail. It really nails down the interaction. So stepping back from the molecular level, what are the bigger picture potential real-world implications of all this?
Well, they're potentially quite significant. Let's start with neurodegenerative conditions.
Like Parkinson's and Alzheimer's.
Exactly. The ability to specifically block BAX-mediated cell death in neurons that could potentially lead to the development of brand new drugs, drugs that might actually slow down or even halt the progression of these diseases.
By preventing those crucial brain cells from dying prematurely.
Precisely. As Professor Grant Dewson from EHI put it, finding drugs that can prevent this neuron loss would be truly game-changing, because right now we just don't have treatments that do that effectively.
That offers a real sense of hope, doesn't it, for conditions that currently have such limited options for slowing them down?
It absolutely does. And then there are the cancer applications we touched upon.
Right, the drug resistance angle.
Yes. The findings suggest a potential new strategy, particularly for cancers that become resistant to drugs like venetoclax because they've lost BX function.
Which happens, you said.
It does. So, WEHI 3773's ability to promote BAK-driven cell death offers a possible way to overcome that resistance, to make those hard-to-treat tumors sensitive to therapy again.
So this one discovery pathway seems to branch out into two really significant areas, neurodegeneration and cancer. Does it also teach us more generally about how cell death works?
Oh, definitely. This research provides really valuable new insights into the intricate ways cell death is regulated, specifically the interplay between BX, BAK, and VDAC2.
Understanding the nuts and bolts better.
Exactly.
Yeah.
And a deeper understanding of these fundamental mechanisms could, uncover even more targets, more avenues for therapeutic intervention in all sorts of diseases where cell death goes wrong, either too much, like in neurodegeneration, or too little, like in some cancers.
Of course, this sounds incredibly promising, but with any research, especially at this stage, there must be caveats, limitations, things we need to keep in mind.
Absolutely, and the researchers are upfront about this. One important point they noted is specificity. While turning off BAX alone might be enough to limit neuron death in the brain,
Yes.
that might not be the case in most other cell types in the body. Other cells might have different dependencies or backup systems.
Okay. So a BAX blocker for the brain might need to be really targeted to the brain.
It suggests that, yes, to avoid unwanted side effects elsewhere, you'd likely need a way to deliver such a drug specifically to neurons, or ensure it only works effectively there. It adds a layer of complexity for therapeutic development.
That makes sense. You wouldn't want to mess with normal, necessary cell death throughout the body.
Not at all. Another thing they found was variability. While EHI 3773 had fairly consistent effects when they used specific drugs to trigger apoptosis,
Right.
its effects on BHI and BAK activity actually varied when the cells were exposed to other kinds of stress or signals that can also induce cell death.
So it depends on how the cell's being told to die.
It seems so. It just highlights again that these cell death pathways are incredibly complex, with lots of different inputs and interacting components. WEHI 3773 isn't necessarily a universal off switch for all apoptosis triggers.
Okay, so it's not quite a one-size-fits-all solution for blocking cell death in every situation?
That appears to be the case. And linking back to the cancer application overcoming venetoclax resistance, that ability relies on the cancer cells having functional BAK protein.
Right. BAK has to be working for DHI 3773 to activate it.
Exactly. So if a BEX-deficient cancer also has faulty BAK, then this approach probably wouldn't work. It wouldn't be effective in all BEX-deficient cancers.
That narrows its potential use somewhat.
It does. And perhaps the most important thing to stress is that this is all still preclinical research.
Meaning not tested in humans yet.
Correct. It's laboratory research, using cell lines and specific experimental setups. There's a significant amount of further research, development, and rigorous testing needed before findings like these could ever become actual approved treatments for people.
So while the science is fascinating and the potential is exciting, there's still a long journey ahead. A lot more work to do.
A very long journey. But the researchers themselves, they seem genuinely optimistic about the significance of these findings.
What did they say?
Well, Professor Grant Dewson, who we mentioned, really emphasized the potential impact specifically for Parkinson's treatment. Professor Guillaume Lessine, another key researcher, expressed real enthusiasm about finally finding a molecule that targets BAX and stops it working in neurons.
That sounds hopeful.
And the lead researcher on the paper, Keming Lee, highlighted that this discovery could pave the way for developing the next generation of cell death inhibitors.
So they see this as a foundational step, a really significant advance in our understanding.
Absolutely. Their overall take seems to be that this study opens up a completely new avenue, a new direction for discovering those desperately needed drugs that can actually modify the course of neurodegenerative diseases.
And so just treat symptoms.
Exactly. And it also provides a strong proof of concept. It shows the principle works for how we might tackle drug resistance in cancer by targeting these fundamental cell death mechanisms involving BAX, BAK, and VDAC2.
It really does make you think about the incredible complexity humming away inside our bodies. Billions of cells making these life and death decisions constantly.
It's mind-boggling, really.
And understanding these processes right down at the molecular level, like this research does, that's how these potentially transformative medical breakthroughs can happen.
Indeed. And if you listening are interested in following how this story develops, it's definitely worth keeping an eye on news and research coming out of places like the Walter and Eliza Hall Institute and the Parkinson's Disease Research Center. They're at the forefront.
It also makes you wonder, doesn't it? This research highlights these intricate, maybe unexpected connections between proteins like BAX and VAC2 in controlling cell fate. What other surprising protein interactions might be out there, just waiting to be discovered, that could hold the key to treating diseases we currently think of as incurable?
That's a really powerful question to reflect on. Where might the next breakthrough come from?
So let's try to bring it all together. This deep dive into the research has really shown us the discovery of this small molecule, WEHI 3773.
Right. A molecule that can block cell death by specifically inhibiting the BAX protein, mainly by stopping it interacting with VDAC2 on the mitochondria.
And this has potentially huge implications both for developing new treatments for neurodegenerative diseases like Parkinson's and Alzheimer's.
And also for finding new ways to overcome drug resistance in certain types of cancer by leveraging the related BA protein.
Our aim here was to explore these sources, the news report and the scientific paper, and really pull out the key information and insights, hopefully giving you a clearer picture of this important cutting-edge medical research.
We hope we've managed to make this complex science a bit more accessible.
Thanks so much for joining us for this deep dive. We hope it's giving you some valuable insights into the forefront of the fight against these challenging diseases.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.